Okay, welcome back for another lecture in the human species. Today's lecture is focused on mechanisms of inheritance. When we talk about inheritance, we're referring to the inheritance of alleles or the inheritance of genes, which code for things that influence the biology of humans. So, in order to understand human biology and human evolution, we have to understand DNA, deoxyribonucleic acid, especially nuclear DNA, which contains the information that parents pass on to their kids. So, when we refer to genes, remember that genes are just stretches of DNA that code for proteins. And so last lecture was focused on DNA, what DNA is, how it's structured, how the functions of DNA are carried out. Those include replication, like making copies of itself when you grow and when you repair the body. And the second one is protein synthesis, right? So structural proteins, hormones, and so many other things that play a role in the body. We know that the process of cell division, or particularly the formation of gametes through the process of meiosis, has importance for understanding the evolution of humans because it increases variation in our population, right? That's why, like, not all kids look like exact blends of their parents. Each kid looks a little bit different, and that's in part due to the process of meiosis. And so remember, through meiosis, we experience forms of genetic recombination, including crossing over of chromosomes and including random assortment. Okay, so today's lectures focus on mechanisms of inheritance. How are things actually passed on from parent to kid? That's a critical part of understanding evolutionary change, not just in humans, but in any animal. Okay, so back in the day, before people really understood biology, they had this idea called preformationism. And the idea is that in each gamete or in each sex cell, they didn't really know what sex cells were. They didn't really understand biology on this level. The idea is that there were tiny versions of fully formed people in individual sex cells. And those individuals were referred to as a homunculus. And so in each individual, we had a bunch of homunculi that were like just little tiny versions of people. That is ridiculous now in our sort of modern understanding of things. But there's a couple of reasons why this doesn't work, why it's problematic. And the first one is that in order for this to be the mechanism of reproduction and inheritance, we'd have to have infinite homunculi, right? So, like, if one individual had sex cells in the body containing these tiny versions of fully formed individuals, those fully formed individuals would also have to have sex cells containing fully formed individuals. And those individuals would have to contain sex cells with fully formed individuals, and so on and so forth through all of time and space, right? So that can't work. It's not possible to have infinite homunculi, separate from the fact that it's just an absurdity to suggest that there are tiny individuals contained in sex cells. The other interesting problem is that if preformationism was correct, it doesn't explain the presence of hybrid animals. So, for example, this is like one kind of classic example where if you have a horse and a donkey and they produce an offspring, you get a mule. You get a hybrid of the two parents. And in many cases, those hybrids, or actually, I guess we could say in all cases, they're not just like carbon copies of the parents, right? They have a combination of characteristics from both. And in many cases, the hybrids are not able to have offspring of their own, right? They're not viable. And so preformationism can't explain how you get a unique combination from two parents, right? If preformationism was correct, then the offspring should be either a horse or a donkey, but it's not, it's a mule. And so eventually, preformationism was replaced by something called epigenesis. And epigenesis is the idea that an organism develops from a fertilized egg, right? So this is epigenesis. And so that's what we now know to be true. Charles Darwin, who we'll spend some time talking about shortly in subsequent lectures, Charles Darwin was a guy, a naturalist who is credited with the formulation of evolution through the process of natural selection. And Charles Darwin thought that traits were passed on through a sort of uniform blend of what their parents had. And so this is the idea of blending inheritance, that there's a uniform blend of the parents' phenotypes, right? The phenotype being the physical expression of the underlying genes that are passed on to offspring. So the idea is that if you have a population of, let's say, cows in this example, and you have one individual that has a variation on coat color, that if this individual mates with, let's say, another individual with a different coat color, that their offspring would be a kind of blend of the parents. The problem, though, is any positive effect, any benefit of having a particular trait in terms of like the survival and reproduction of the individual would eventually just be blended out by the other parent. And so this blending out or diminishment of those traits continues over subsequent generations. And so eventually, right, from one generation to another generation, to a third generation, to a fourth, and so on, eventually this original variation, this original difference in coat color, by the end would just be blended out. And so some traits that we observe in living things appear to blend, but they're not actually like a true blend in this kind of way. And so we'll come to learn that variation in a population, the fact that not every individual is the same, is a critical part of evolution. It's how evolution works, particularly through something like natural selection. And so under Darwin's blending inheritance, variation in populations can't be maintained. And so this is actually like one of the things that people were critical of Darwin for, which is that he didn't understand, and nobody understood, how traits were passed on from parent to offspring in the 1800s, which is when Darwin was publishing on evolution. Okay, so eventually, the concept of blending inheritance through somebody like Charles Darwin was replaced. It was replaced through something called particulate inheritance. Particulate inheritance. The idea that there's a transmission of something from a parent to their offspring that contains information about their phenotype. Right? In other words, parents pass on factors from generation to generation, which are not easily observed. And so these factors were discovered by a guy called Gregor Mendel, who was an Austrian monk, and who we now know as sort of like the, quote, father of genetics. I always find it kind of strange when people make discoveries and we call them the father of genetics. It's just a kind of weird, like, cultural tradition, I guess. It's a kind of strange thing. Anyway, so Mendel is known as the, quote, father of genetics, and he discovered the transmission of factors that were passed on from parent to offspring through his experiments that he conducted on the common garden pea. And so In this time, Mendel and his contemporaries, the other people like working on these kinds of things, didn't know what DNA was. They didn't know how traits were passed on from parent to kid. Mendel, through his work, didn't really discover DNA. He still didn't really know what DNA was. He didn't really know what genes were. Um, it was only until the 1900s that his work was kind of rediscovered by William Bateson, and the term genetics was coined. And so there's a story. I don't know if it's true actually. I should probably find out. But there's a story that Charles Darwin had some of Mendel's work like in his workspace at his house or whatever. So maybe Darwin was reading about the transmission of factors, which we now know as genes and DNA, even though he never really discussed it in his writings. Okay, so Mendel kind of lucked out in breeding the common garden pea plant because there's variation among these plants, right? They don't all look the same, and that variation is very simple. It's what we would call dichotomous variation, right? So either like they have one trait or another, right? So either the plants have green seeds or yellow seeds. Either they are, let's say, tall or short, right? It's dichotomous. It's one or the other. The other important thing is that the traits appear to be relatively like independent from one another, right? So you can have green seeds or yellow seeds and be tall or short, right? You can have round seeds and wrinkled seeds and have green seeds and yellow seeds, right? So you can have all these different combinations. And so these plants were so-called like true breeding plants, which means that they could be bred and produce strains that resembled the parental generation time and time again. And so these are some of the characteristics or the traits that Mendel focused on, right? So different characteristics of seeds, different characteristics of the pods for the pea plants, different characteristics of their flowers, of their stems, and so on. Okay, so basically what he did was he started one generation. This is like the true breeding parental generation, and he would breed two variants, right? So in this case, he bred tall variants, we use some of the tall pea plants with some of the short pea plants. And it's important to keep in mind when thinking about his experiments in breeding, he's not just breeding like one plant with another plant. He's breeding a bunch of plants with a bunch of other plants, right? So in other words, we have like a group of tall plants and a group of short plants. Okay, so that's the first step. He did that, bred tall plants with short plants. And the outcome in this subsequent generation, in this generation of offspring that derives from the parent generation, all of the plants would be tall. 100% of the plants bred from all of the short plants and all the tall plants would be tall. So we would consider this the F1 generation, the first generation from the parental population. Okay, then he bred plants, a bunch of plants from this generation. So all tall plants would breed in this F1 generation. And lo and behold, the short trait would reappear, it would reemerge. And he found a consistent pattern in the F2 generation. Again, F2 being the generation that comes from all the individuals bred in this F1 generation. In the F2 generation, he observed the same pattern over and over again, which was that 3/4 of all the plants of the F2 generation would have the phenotype of the F1 generation, right? In this case, they'd all be tall, or 3/4 of them would be tall. And 1/4 of the plants in the F2 generation would have the other trait, that disappeared in the F1 generation. So 3/4 would be tall, and the remaining 1/4 would be short. So 75% tall, right? We can just think of this as 3/4 of a dollar, right? 75% would be tall, and the remaining 25 would be short. And so this trait or this phenotype that disappeared in the F1 generation and reappeared in the F2 generation would become to known as the recessive phenotype or the recessive allele. Whereas the other trait that was demonstrated at 100% in the F1 generation and which occurred at 75% in the F2 generation would be called the dominant trait or the dominant allele. And so the dominant one is an allele. An allele or allele is just a variation of a gene. It's a gene that's always expressed when it's present in the genotype of an individual. The recessive allele or the recessive gene is one that's masked when the dominant allele is present in that individual. So if you remember, if you think back to the human karyotype and the fact that in human somatic cells we have 46 paired chromosomes, right? 23 pairs. For each pair, you get one chromosome from mom and one chromosome from dad. And so if on each of those chromosomes at a particular location where the gene for tall plant height is found or short plant height is found, if I have a dominant allele or a dominant gene there, no matter what the other one is on the other chromosome, I'll be a tall plant. If I have two recessive genes, one from mom, one from dad, then guess what? I'll be a short plant. Okay, and so we know from last lecture that genotype just refers to the genetic makeup, the underlying genes, the underlying alleles that an individual has. The phenotype, on the other hand, is the physical expression of those underlying genes. Okay, a locus is the location of a gene on a chromosome. So I remember locus as location, right? They start with the same three letters. So a locus is a location of a gene on a chromosome. Remember that chromosomes are just coiled strands of DNA. And so basically, like, if you took one chromosome and you pulled it apart, you would have a stretch of DNA. You would have a series of nucleotides, which, as we know, contains nitrogenous bases. So as DNA, it's either going to be, the nitrogenous base will either be adenine, thymine, cytosine, or guanine, right? A, T, C, and G. And so this stretch of DNA from one end to the other will just be a series of nitrogenous bases. When they're coiled together and condensed together, they become chromosomes. But they still have the same sequence of A's, T's, C's, and G's. And so the particular location or the particular spot in the sequence of A's, T's, C's, and G's from one end to the other is the locus. It's the location where we find genes on particular chromosomes. Okay, and so an allele, as I've mentioned, is just a variation. It's just a form of a gene, right? So you can have genes for eye color, let's say. You can have different genes, different alleles for eye color, right? So blue eyes have certain alleles, brown eyes have certain other alleles, and everything in between. Okay, so remember again, for each of the 23 pairs of chromosomes that we have in every somatic cell in our body, we get one from mom and one from dad, right? One is maternal from the biological mother, and one of them is paternal from the biological father. And so on each of those chromosomes, on each of those pairs, you inherit a gene at a particular locus from the paternal chromosome and a gene at that same locus on the maternal chromosome. So, for example, if there's a gene that in our pea plants that codes for the height of the plant, each plant will have two copies of that gene, one from the paternal side, one from the maternal side. If by chance that individual inherited two recessive copies, two recessive alleles, which in this case would be the short allele, on one or on both of those chromosomes, then that individual would have the recessive phenotype. Okay, so in terms of gene expression, there are two sort of conditions. The first is called homozygous, where you've got the same allele at both locations, at both loci of a gene. And so we can refer to homozygous, the homozygous condition as HO, homozygous, the same allele. The other condition is heterozygous, which refers to two different alleles at those locations, at the loci of a gene. And so loci is just plural for locus. Loci just means locations. All right, so if you've got a gene with two alleles, two variants that you can inherit from your mother or father, there are then three possible combinations that you could have. You could, let's say, in this case, the alleles are the blue allele, which stands for some trait or for some phenotype, and a green allele, which stands for a different one. So you could inherit two blue alleles, two of the same alleles, at a particular locus for a gene. And if you have two of the same alleles from maternal and paternal contributions, then that individual would be homozygous for those alleles. If you inherit one blue allele and one green allele, you would be heterozygous at that location. And the final possibility is inheriting two green alleles, again, one from the paternal side and one from the maternal side. In that case, you would be homozygous green. If, in this hypothetical scenario, there were three versions of a gene, three different versions, three alleles, then that means there are now six possible combinations, right? You can be homozygous for blue, homozygous for green, homozygous for purple, the new allele, the new variation, or you can be heterozygous blue-green, heterozygous purple-blue, and heterozygous purple-green. Okay, so we have the genotype, that's the underlying genetics, the underlying genes or alleles that you inherit from the maternal and paternal side. And we have the phenotype, the physical expression of the genes that we have. In this case, here's our example with the tall and short varieties of the pea plant. So, in this case, we have three different genotypes because there are two alleles, there's two variations, tall and short. And so T, big T, capital T, is the tall allele. And if an individual has two capital T's or two dominant alleles, they are homozygous dominant, big T, big T. If, on the other hand, they have two lowercase t's, they are homozygous recessive. If they have a mix, if they have a combination of both, they are heterozygous. They get one capital T, one dominant T from mother or father plant, and one recessive or lowercase t from mother or father plant. And so if they have one capital T, whether they're homozygous dominant or heterozygous, the phenotype is always tall. because that allele will always be expressed no matter what the other copy is, no matter what the other allele is on the other locus on the paired chromosome. The only way the other phenotype is expressed, in this example, the only way that the short phenotype is expressed is if the individual inherits two heterozygous, or excuse me, two lowercase alleles, right? Two short alleles. And that's because in the heterozygote, the expression of this allele is masked by the presence of the other one, by the presence of the dominant allele. So in other words, the only way for this recessive allele to be expressed is if there isn't a dominant allele there to block it, to mask it, to prevent it from expressing. So in this case, you only see the short phenotype when you have two of these recessive alleles. And of course, if you've got two of the dominant alleles, again, each of which comes from the maternal and paternal plants, then obviously the dominant phenotype is expressed. Okay, so from Mendel's work on pea plants, he sort of figured out that there were these factors that were passed on from generation to generation. He didn't know that the factors were genes. He didn't know that they were genes at a particular locus on the chromosomes that we inherit from our parents, from our biological parents. And so from his work, he developed three different sort of laws or principles. And so this is one of them. This is Mendel's principle of segregation. And the idea is that the two alleles of a gene found on those pairs of chromosomes segregate independently of one another into sex cells. And so because of this, we can look at the possible combinations of alleles preserved in parents. So let's say this is the maternal side, and this is the paternal side. You can get, let's say this is an easy example, because both of these parents, maternal and paternal, are homozygous. They have one, in this case, they have the same alleles at both location on the chromosomes. And so basically through the production of their sex cells, right, through the production of eggs on the maternal side and the production of sperm on the paternal side, basically in each sex cell they get one of these chromosomes. And so through the process of meiosis, through the formation of gametes or sex cells, you get sex cells that are haploid, that have 23 unpaired chromosomes. And so in this case, each gamete can only have a blue allele on the maternal side in this case, and a green allele on the paternal side. If each parent was heterozygous, right, like if there was a blue and a green on each of them, then their gametes could have any one of those, right? Each gamete, each egg produced on the maternal side could have a blue or a green allele. Each sperm on the paternal side could have either a blue or a green allele. And so because of that principle of segregation, if we know the genotype of each parent, and by the way, I'm referring to maternal and paternal because that's how we think of humans. Plants are kind of wild and crazy on the biology and reproduction side, which leads to cool like evolutionary outcomes. But I'm talking about plants in this way, in the sort of animal way, because it helps us apply it to humans. Okay, so because of the principle of segregation, if we know the genotype of maternal and paternal contributions, then we can sort of figure out what the possible offspring genotypes are through looking at the possible combinations of each allele from maternal and paternal sides. So, for example, if one parent is a tall plant and has the genotype homozygous T, so two dominant Ts, and if the other plant has the short phenotype, which means that its genotype is homozygous recessive, we can create what's called a Punnett square. And some of you may have encountered Punnett squares in biology class previously. So if so, this will be familiar to you. If not, it looks like this, where the each of the alleles are listed on one side. Let's say this is like the maternal side, and let's say that this is the paternal side. So each allele basically gets its own location, its own cell in the Punnett square. Each of these gets its own location or its own cell in the Punnett square. And then we can just combine alleles for each of them. And so basically what we see is if you've got two parents with these genotypes, the only possible offspring genotypes are heterozygous, right? So in other words, if you bred 100 plants with this genotype, the homozygous T, big T genotype, and 100 plants with the homozygous little t genotype, 100% of the offspring, all of the offspring, would have this genotype. It's the only possible genotype. And so this is the parental cross that Mendel performed. That was his first step, where he bred true breeding tall plants with true breeding short plants, and found that, guess what, 100%, 4 out of 4, in this Punnett square example, of the offspring had the same phenotype. They were all tall. And remember, because in each of these genotypes, there's a dominant allele and a recessive allele, the dominant one is always going to be expressed. The recessive allele can't be expressed because it's paired with a dominant one. So in other words, 100%, 4 out of 4 of the offspring have the tall phenotype. That's the result that Gregor Mendel attained through his first experiment, his first step in these experiments. If we then cross two individuals from this generation with this heterozygous genotype as part of the F1 cross, again, we have the dominant allele and the recessive allele for both parents. Now we have different combinations for the offspring genotypes. We can have big T, big T, big T, little t, big T, little t, and little t, little t. So now we have 3 different genotypes, 2 homozygous genotypes. Right, here's a homozygous, and here's a different homozygous genotype, and 2 of the same heterozygous genotypes. So of these 4 possible genotype combinations, how many of those have the tall phenotype? Take a second to think about it. Of those four possible genotype combinations, how many will be tall? Okay, now think about, of those 4, how many of them will have the short phenotype? Okay, hopefully you figured out that there will be 1, 2, 3 tall plants and 1 short plant. So in other words, for the tall phenotype, 3 out of 4, or 75%, will be tall, and the remaining 1 out of 4, or 25%, will be short. And that's exactly what Mendel found when he crossed plants from the F1 generation. the recessive phenotype, the short phenotype associated with this homozygous recessive genotype reemerged. It reappeared in the F1 cross. Okay, here's another one of Mendel's principles. It's the principle of independent assortment. Okay, remember that one of the advantages of performing these experiments on the pea plant is that the traits of interest appear to be independent of one another. In other words, that like offspring can inherit all different combinations of traits. In other words, it's not the case that if the plant is tall, it always has yellow seeds, or if the plant is short, it always has green seeds, or if the plant's tall, it only has wrinkled seeds, or if the plant's short, it only has round seeds, right? Each offspring can have any combination of the traits associated with the parents. And so here's a Punnett square which looks at two traits together instead of just one. So in this case, this is looking at one one trait with two alleles. This is looking at two traits with four alleles. So in this case, the traits are the seed color, right? Like one trait is what seed color the plant has. Is it yellow or green? And the other trait is whether the seed is round or wrinkled. So two traits with four different alleles, right? There's the big G allele for the yellow seed. There's the little g allele for the green seed. There's the big W allele for having a round seed shape, and the lowercase w for having a wrinkled seed shape, right? So two traits with four alleles, two alleles for each of the traits. And so here are the different possible parental genotypes, right? In other words, homozygous, dominant and homozygous recessive, or heterozygous, homozygous dominant, homozygous recessive, or heterozygous. And basically you just like with a single trait Punnett square, you do the same thing. So in this case, the genotype is big G, big W, and that translates to having a yellow seed because of the dominant alleles there, and a seed that's round. And so because genes on different chromosomes are sorted into sex cells independently of one another, all of these combinations are possible. So yellow round, yellow wrinkled, green round, and green wrinkled, right? Every combination is possible. We could calculate genotype and phenotype frequencies for this Punnett square just like we might do for the previous one. And so in this case, the genotype frequency would be 1 out of 4 is homozygous dominant, 1 out of 4 is homozygous recessive, and 2 out of 4 are heterozygous. So if you add all these up, 1 plus 1 is 2, plus 2 is 4, you get 4 out of 4 different genotypes. We've already talked about the phenotype frequencies, right? 3 out of 4 are tall, and 1 out of 4 are short. We can do the same thing with this more complex example. These are all of the different genotypes, and each of these genotypes are found in the Punnett square, right? So here's this genotype there, here's this genotype here, here's the third genotype, and so on. And so you can go through and count these up and see, oh, okay, these are the same, these are the same, these are the same, and so on, to calculate what are the frequencies of each genotype. We can then look at the frequencies of each phenotype. So, like, in other words, how many of them are yellow and round? So we could count them. One, whoops, 1, 2, 3, 4, 5, 6, 7, 8, 9. So 9 of the 16 squares, 9 of the 16 possible phenotypes are yellow and round. So it's 9 out of 16. We could do the same thing with yellow and wrinkled, with green and round, and with green and wrinkled. And so hopefully you can see with the phenotype frequencies, when you look at this, the highest numbers are associated with dominant phenotypes. So remember that the yellow color, which is associated with big G, with that particular allele, is the dominant allele. And in terms of seed shape, the uppercase W is the round, is the dominant allele. Whereas the green seed color and the wrinkled seed colors are the recessive ones. And that's why to get offspring that are both green and wrinkled, it's going to be rare because they're both recessive alleles. So this phenotype will be the rarest of them all at 1 out of 16. Okay, so many traits are not so simple. When we're thinking about human traits, they're affected by hundreds of genes. And so you can imagine doing Punnett squares for hundreds of genes interacting with one another in a non-independent way would be incredibly complicated. Okay, so we talked about 2 of Mendel's principles. Remember, the first one is the principle of segregation, which stated a different way is that you have 2 copies of each gene. You get one from the biological mother and the other from the biological father. The principle of independent assortment states that these 2 genes that you get could be the same alleles, or they could be different, depending on what your parents have themselves. So if they're the same alleles that you get, then you'll have then for that particular trait, you'll be a homozygous individual. If you have 2 different alleles, you'll be a heterozygous individual. And the third principle, which we've talked about but we didn't name as a principle, is the principle of dominance, which is that these alleles together determine the phenotype. If you have a dominant allele, it will always be expressed. If you have a recessive allele, it can only be expressed in the presence of a second recessive allele. It's masked by the presence of a dominant allele. Alleles can also be, like simple Mendelian traits, the alleles can be also something called codominant, which we'll talk about in just a moment. Okay, so let's go through another example of a Punnett square of a simple trait with 2 alleles. So the trait is hamster coat color, and the alleles are a black coat color allele and a white coat color allele. And we could say that the black coat color allele is associated with the capital B, uppercase B, big B, and that's dominant, whereas the other allele is recessive, lowercase b. So if we have a parental generation with 2 parents that have different coat colors and different genotypes, we can breed those 2 parents and observe the outcome for their kids, for their offspring. And so one parent has the genotype homozygous dominant, big B, and the other parent has the genotype homozygous recessive, little b. And so if we combine alleles from both parents to figure out what are the possible combinations for the kids, what are they? Take a second and do this yourself. Okay, hopefully you know that each of the possible offspring combinations, it's all heterozygous. Big B, little b, and so on. What is the phenotype for each of these genotypes? Take a second and think about that. What's the coat color for each of those genotypes? Yeah, it's the black coat color. And so this is the same thing that we observed in Mendel's process, where you've got a parental generation. We can just call this P. You have the first generation after that crossing, the F1 generation, and 100% of those individuals will have the dominant phenotype, will express the dominant phenotype. So in terms of genotype and phenotype frequencies, there's only one genotype and there's only one phenotype. So there's 4 possible combinations, 1, 2, 3, 4, and all 4 of them have the same genotype and phenotype. So in this case, they're both present at 100% in this generation. We can then cross individuals from this F1 generation. And again, all of those individuals have the same genotype. They're both heterozygous. So take a second to think about and write down the possible genotypes and the possible phenotypes for this cross. And I'll give you a few seconds to do that. Okay, hopefully you came up with this result, right? This genotype combination will be homozygous dominant. This one will be heterozygous. This one will also be heterozygous. And this one will be homozygous recessive. And so now, in the F2 generation, we have a different pattern emerging. We now see the reappearance of this trait of the white coat color that we first observed in the parental generation. And that's because in order for that coat color to be expressed, we need to have the genotype homozygous recessive. And that genotype wasn't possible in this first, this F1 generation. Okay, so now we have 3 different genotypes and 2 phenotypes. And so we can calculate the genotype and phenotype frequencies. In this case, the homozygous dominant genotype only occurs in 1 out of the 4 possible combinations. And so that genotype has an occurrence of 1 out of 4, 25%. 25% of this generation, of the F2 generation, will have that genotype. For the homozygous genotype, we have something different. We have 2 occurrences. And so 2 of the 4, or 50% of the F2 generation, will have that genotype. And finally, for the homozygous recessive genotype, 1 out of 4, or 25% of the population, will have that genotype. For phenotype frequencies, it's a little bit different. These 2 genotypes, again, because there's a dominant allele present, will have the black coat phenotype. And so, of the 4, you've got 1, 2, 3 individuals with a black coat. So that's 3 out of 4, or 75%. So 75% of the F2 generation will have a black coat, 3 out of 4, and 1 out of 4 will have a white coat, or 25% will have a white coat from this generation. And so again, this is the same pattern that Mendel found when looking at tall and short plants. You have a parental generation with two different traits, two different variants, or two different forms. You have an F1 generation where 100% of the offspring have one of those forms. And then in the F2 generation, this form reappears. And it's all a product of how the underlying genotypes are produced through passing on of alleles from parent to kids, and how recessive phenotypes are expressed only if individuals have the homozygous recessive genotype. Okay, so what about this thing called codominance that I mentioned? Well, in this case, neither allele is dominant. The alleles are expressed at the same time. They're co-expressed. And one example of this is the ABO blood system. So we each have a blood type. And it turns out that depending on the alleles that we inherit from biological mother and father determine what our blood type is. And so this is an example of one of a few, so-called simple Mendelian traits where we can treat this like the pea plant. We can treat this like the hamster coat color. So let's say, so I'll start by explaining the different blood types here. Let's say one parent has blood type A, and that's because their genotype is AO, and the O allele is recessive. In this case, we have three alleles. We have an A allele and a B allele, and both of these alleles are codominant. Neither one is dominant to the other. The O allele, the third allele, is recessive. And so this is expanding our pea plant example and the hamster coat example in two ways. One, there are three alleles, where in the previous example there were only two alleles, tall versus short, black coat versus white coat. Right now we have three alleles, the A-type allele, the B-type allele, and the O-type allele. The second way that this is different is the A and B alleles are codominant. Neither one is dominant to the other. Okay, so one parent has the AO genotype at this particular locus on this pair of chromosomes. One gene is the A allele, and the other gene is the O allele. Their blood type is therefore A, because O is not expressed, it's recessive. It can only be expressed if this other allele at this locus was an O allele. Then they would have blood type O. Okay, the other parent has blood type B, that's their phenotype. Their genotype is BO, because at this locus, on the same chromosome pair, they have the B allele, and at the same locus on their other paired chromosome, they have the O allele. And so if these two individuals have kids, let's say that they had 100 kids, which is essentially impossible. But let's say, for sake of our argument, they had 100 kids. One possible combination of alleles is they get an A allele. Let's say that this is dad and this is mom. They get the A allele from dad and the O allele from mom. Guess what? They have blood type A with the genotype AO. Let's say another offspring, another kid, gets the A allele from dad and the B allele from mom. Their blood type is now AB, because A and B are co-expressed. They have neither blood type A nor blood type B. They have blood type AB. A third combination is they inherit the O allele from dad and the B allele from mom. And so now their blood type is blood type B, like their biological mother, and their genotype is BO, like their biological mother. The fourth combination is they inherit the O allele from dad and the O allele from mom. And so now blood type O can appear because both of these alleles are recessive, they're not masked by the A or B allele, and this kid has blood type O. Now, I mentioned, like, let's say they had 100 kids, because this is like a probability. If they had one kid, what's the chance that they inherit any one of these? So when we're looking at pea plants, when we're looking at hamsters, when we're looking at blood types in humans, we're treating this like a sample. We're treating this like if they had 50 kids or 1,000 kids or 10,000 kids, these are the different combinations. These are different possible combinations. But if they just had one, they could have one kid with any one of these. If they had 2 kids, they could have 2 kids with the same genotype. They could have 2 kids with different genotypes, right? It's like a roll of the dice. Okay, so that's codominance. Most traits, however, are not simple Mendelian traits. In other words, there aren't like one gene or a couple alleles that code for one phenotype. It's not like, oh, I got the gene for tall height, I'm now a tall person. Oh, I have the short gene, I'm a short person. No, actually, most traits that we care about that have implications for human biology, for health, for human performance, most of those traits are complex. They are complex traits, not simple Mendelian traits. And they're complex traits because they're not dichotomous. In humans, for example, if we think about the height case, we don't have tall and short humans. We have a range of human heights that varies continuously from the low end to the high end. And so usually complex traits are traits that are have a distribution, a continuous distribution. In this case, they have a so-called bell shape, where if you plot the distribution of that particular trait, if it's a quantitative trait, most individuals will be somewhere in the middle of that range. Some individuals, fewer individuals will be at the low end, and fewer individuals will be at the high end. This is a really cool kind of like visual where they asked men and women to line up according to what their height is. So here's 5 feet 0 inches, 5 feet 1 inch, all the way up to 6 feet 5 inches. So they said, like, okay, in this case, the individuals wearing the white shirt and hat are women, and the other people are men. And they said, okay, line up behind whatever sign characterizes your height. So I'm 5 feet 11, I would line up here. And what you see is that naturally, most people, most men and women are going to be somewhere within this window. Because there's an average or mean difference in height between men and women, most male individuals in this sample of people are 5 feet 10, right? If you count each individual, most individuals here will be this height. For the women in this sample, most of them will be 5'6", right? Most individuals are here in this line. And you can see that for the men, this is the shape of the distribution. For the women in this sample, there's a different shape of the distribution. But for both of them together, they have this bell shape. And we could say that like the peak of this distribution is in a normal distribution, this is the average. But the interesting thing from like a math and stats perspective is that this sample average is lower than the male average and higher than the female average. It's somewhere in between, right? Because it's pooling the sample together, pooling like POOL, not pooling like pull the door open. Okay, so many traits are going to exist on this kind of scale, and Usually, there's an environmental component to the expression of the underlying genotype. So, with the pea plants, with the ABO blood system, with the hamster coat color, we didn't look at this. We just assumed that whatever the genes were, whatever the alleles were, that determines the phenotype. Right? If you're a plant and your genotype is little t, then guess what? You're going to be a short plant. But for most complex traits with many, many, many alleles, hundreds of alleles that are all interacting, and with the filter of the environment, it means that we're going to get many more possible outcomes, many more possible phenotypes. Again, ranging from 5 feet to 6'5", or 4 feet to 7 feet, or whatever it might be. Okay, so we can look at the effect of the environment on height as phenotype. And so this is on the y-axis, height of boys in centimeters. And on the x-axis is their age, how old they are in years. So 5 years old, 10 years old, 15 years old, 20 years old, and so on. And so what we see just in general is that as you age, you get taller. You grow up to be taller, usually. So in other words, 5-year-olds are shorter than 15-year-olds. That makes sense. As you get older, your bones grow, you get taller. Everybody grows at different, in different ways, at different rates. Some people start out shorter and grow to be taller. Some people start out taller and don't grow as much. But in general, we observe this relationship. That relationship is related to your biology. It's related to your genes. Your bones grow, you get taller. That's a genetic thing, in part. However, there's also a strong effect of your environment, like your stress and your nutrition on your body's ability to grow and to produce a taller height. And so this is the difference in laborers, kids that had to work from a young age, to non-laborers in the year 1874. So this is before like child labor laws where kids could have jobs, right? 7-year-olds can't work a hard job. They can't work any job now, right? Because we protect kids from that, unnecessary stress. And so you can see that the growth rate, right, how much height changes over time for laborers and non-laborers is about the same. However, the non-laborers, if you follow this back, if you follow both of the lines back, the non-laborers are taller than the laborers. The non-laborers are not under the same kind of stress. They're able to have increased nutrition compared to the laborers, and that influenced their height at young ages. If we look at wealthy populations compared to average populations from the 1950s, like this is the average population height or the average sample height in the 1950s. The other line is the wealthy height in the 1950s. You can see a difference there too, right? In other words, the red line is above the other line, which means that their height is increased. So this difference in height, through age from 5 to 20 or whatever it is, from laborers to non-laborers in 1874 to average kids and wealthy kids in the 1950s, is not something genetic. It's not that like wealthy kids in the 1950s have genes for being taller. It's just that they have access to better nutrition and less stress, like less environmental stress on the body due to their wealthy environment. And that influences their phenotype. That is the environmental component influencing how tall they are, not what gene they have. Um, so there you go. Better nutrition and health as part of the environment leads to increased height with no genetic difference. Right? They're not taller because they have a gene for being taller. They're taller because their body is able to grow to their genetic potential because they're not stressing their body and because they have enough calories and nutrition to grow. Okay, here's a different example where the environment isn't external, it's internal. And so this is an example, a common example that we know has a major effect, which is something like a sex hormone like testosterone. So we can look at the relationship between arm strength and age in biological males and females. And so at young ages, there's no difference between males and females. As you grow to be uh, larger as you age, your arm strength increases. You get bigger and stronger. But um, after about 13, between 13 and 14, there's a big division between biological males and females in terms of the development of their arm strength according to this data set. And that's linked to the production of increased testosterone around the time of puberty. So in biological males, testosterone increases, arm strength continues to increase, whereas in females, there's a different pattern because females don't have the same degree of testosterone production as biological males do. And again, it's important to emphasize the fact that these lines are average lines, right? This is an average. Some individuals will be here, other individuals will be there, both within each biological sex, right? So some males will have, crazy increased testosterone development. Other males will have less testosterone. Some females will have increased testosterone. Other females will have even less. And so these are averages, right? And so on average, this is the difference here. And so if you look at like different measures of performance in biological males and females, there are differences when we're thinking about things that are related to strength. And that's related to this underlying internal environment. And so here is some data on Olympic records for javelin throwing. And in females, biological females, it's 71.7 meters. And in biological males, it's 98.5 meters. And again, that's because this is related to strength. If you look at things that are not related to strength, something that's related purely to skill, there's like let's say archery. There's no difference in biological male and female performance. Again, because this has nothing to do with strength. So the point is, the environment can be external, right? It can be the kind of nutrition that you have access to. It can be the stress that the body, like the physiological stress the body is experiencing due to external factors, like what kind of job you have, what kind of place you live in, your socioeconomic status, etc. But it can also be internal. It can be the internal environment or the development of things like sex hormones, like testosterone, through your growth and development. And so all of these things interact. Your underlying genes, and in many cases, hundreds of genes that influence a particular trait, the environment that you experience through your growth and development, internal and external, both, which all interact to produce the phenotype. So if you look at this example, where you've got three different genes, gene 1, gene 2, and gene 3, each of which have different numbers of variants, each of which have different alleles, right? So gene 1 has three different versions, three different alleles. Gene 2 has these two alleles, and gene 3 has these four alleles. So you can think of like hamster coat color, black or white, ABO blood group, ABO alleles. Let's say gene 3 is, I don't know, hair color, really dark, a little bit less dark, a little bit less dark, very light, let's say, hypothetically. You can then have these different genotype combinations. right? If you only have two alleles, red and not red, you can be homozygous red, homozygous not red, or heterozygous. If you have three alleles, then you have 6 combinations. If you have 4 alleles, you have many more combinations, and so on. Let's say you have these 3 different environments, A, B, and C. Again, these can be external environments or internal environments. The environment, as we've been mentioning, acts as a filter on the genotype that you have. So let's say for each of these genes, this individual has this genotype, and this genotype, which is filtered through environment A, which results in a particular phenotype. Okay, you can then have a second individual, second person, with, guess what, the same genes, the same underlying genotype. But this time, they grew up in environment B. And again, this could be a biological male or, versus a biological female's developing body in terms of the internal environment. It could be different historical times when maybe kids were allowed to work hard jobs at the age of 6 or whatever it is. Different environment in this case, but the same genes can lead to a different phenotype. And again, the phenotype can be anything. It's not just height or eye color or whatever. It's any biological trait that ultimately is linked in some way to underlying genetics. So these 2 individuals have the same alleles, the same genes, but a different phenotype because of the environmental effect. We can have an individual with different genes, different alleles, and a different environment, which leads to different effects, and so on. And so we can go through this entire example, and come up with 540 possible different phenotypes just based on this combination of genes, alleles, and environments alone. And so when you expand this out to include hundreds of genes and hundreds or thousands of different kinds of environments, both internal and external, you can imagine the phenotypic complexity, the complexity in human traits or any animal's traits, right? Again, think about height. It exists on a continuous scale and is influenced by 700 genes. Okay, so what about evolution? Well, evolution happens when genes or alleles in a population change over time. And this takes thousands of years. And so new alleles can pop up through mutation, and existing alleles can become more or less common. But it's important to know, when we're thinking about evolution, we define evolution based on genes, right? I just said evolution is change in alleles over time. But we know that most complex traits have a strong environmental component. And the environment can change over time. And so it's important to know that, like, when we're thinking about traits in humans and we're thinking about the evolution of human traits, that we're not just talking about genes. We're also talking about the environment. And so this is why you should be skeptical when you see a news story or you read a paper or you see or you hear people talk about a gene for this, a gene for that, right? Like a gene for being really smart or a gene for musical ability or something like that. It's not that simple. It's not that simple because the environment filters the underlying genetics. It's not that simple because most traits are influenced by hundreds of genes, not just one. Most genes are not like simple Mendelian traits, not like the pea plants. Okay. We said this already. Evolution occurs through change in alleles in a population over time. We'll discuss what evolution is in more detail next time, but that's our basic sort of like starting point definition. Okay, so let's think about how evolution might work just in a really simple way based on our height example. So again, here's our distribution of heights in this sample of people. They said, hey, line up behind the sign that describes what your height is, right? Bunch of males lined up here, that's just below 6 feet. Bunch of females, individuals lined up here, that's just around 5'6" for this sample, right? Not for all humans, but for this sample of individuals in this picture. What would happen if someone with tall alleles and with a very tall phenotype was added to the picture? What if a bunch of tall people showed up? Where would they go? Where would they stand in line? Over here, right? How would the distribution of alleles in this population, how would the gene pool change? How would the percent or the frequency of different genes or alleles change? It would probably increase the frequency of different alleles related to being taller. So allele frequencies might change. And guess what? If alleles are changing, that's a form of evolution. Now, this hypothetical example where you tell some new tall people to line up with this group, that's not really evolution, right? But we're thinking about evolution in terms of the entire species. But we can use this as a kind of toy example. How would the average height change if tall individuals got added to this side? Well, it would shift. If this is the first generation, and after 100 generations, if more and more tall individuals were added, the average would shift to the right. In other words, the peak, again, like let's say we can draw the male distribution, the peak is now here. Whereas in this case, the peak was here, let's say a little bit below 6 feet. The peak after generation 100 with adding more tall individuals would be like 6'4". The peak for the female group would be like 5'9" compared to a peak of 5'6". So that's an example of directional evolutionary change in the direction of taller heights in this scenario. Now, it would be evolutionary if adding tall individuals was related to their survival. If they were, quote, added through the fact that taller individuals were surviving at higher percentages than shorter individuals, then the population over time would become taller because they would pass on those tall alleles to their kids. Okay, now what happens if only short people could have kids? That the tallest individuals among the biological males and the tallest individuals among the biological females, for whatever reason, were not able to have kids because of their height. So now, only the shorter individuals are reproducing and having kids. How would the gene pool change? How would the frequency of their genes change generation after generation? Well, you wouldn't have tall alleles because they can't have kids. And so average height would decrease. The frequency of shorter alleles would increase, and evolution would occur. You'd have another example of directional evolution, but in the opposite direction after hundreds or thousands of generations. And so, in short, that's how directional evolution works. But you're probably still thinking, yeah, but what does this have to do with like having kids, and how do you add shorter people or tall people or whatever? Yeah, well, I'll explain that in more detail next time. Essentially, if we're talking about natural selection, which this is kind of a quasi example of selection, it's linked to survival and reproduction. That's called fitness. And so I'll explain that next time. All right, let's talk about the data portion for our first lab. Okay. Lab 1 is looking at the effects of genes and environment on phenotype, right? The stuff that we've been talking about the last couple of lectures. And we have 3 questions, and the first lab worksheet that's due today is asked you to come up with 3 hypotheses, or one hypothesis for each of these 3 questions. So the first one is, how does parent height influence offspring height? Is offspring height tightly linked to genetics, or is some of the variation in height of the offspring related to environment? Just a hint, we talked about some of that in this lecture. What is the effect of temperature, measured as latitude in degrees on the planet Earth, on limb proportions? And by limb proportions, we really mean the relative length of the lower limb. I mentioned last lecture that in humans and animals, we observe something called Bergmann's and Allen's rules, where in cold climates, we tend to see shorter, stockier species and populations. And in hotter climates, we tend to see taller individuals with longer legs and longer limbs in general. And that's related to maximizing volume in cold climates, which volume basically is involved in heat production and retention, versus maximizing surface area in hot climates. And the more surface area you have, the more heat dissipation you experience. And then number 3, how do body size and proportions affect grip strength? And these are like 3 different traits, just looking at height, looking at temperature and limb proportions, and looking at strength, grip strength. We talked a little bit about that in this lecture as well. Okay, so for each of these, for lab 1.1, again, due today on Blackboard, you have to come up with a hypothesis or prediction for each of them. Next lecture, we'll discuss these different questions and some of the hypotheses that you came up with. Okay, so let's break down that first question. How does parent height influence offspring height? And what's the impact of the environment? So we have like 3 things right here: height and environment. Well, to test hypotheses in science, we analyze data. We don't just guess, because that tells you nothing. An educated guess is a hypothesis. But in order for us to do science, in order for us to uncover some truth about the world, we have to analyze data. Because science, remember, is empirical. Okay, so what do we mean by data? Data is actually the plural for datum. Datum. And a datum is a piece of information in some way. So we need many pieces of information on height. Data then is the plural. Data refer to more than one piece of information. Another definition I saw is facts and statistics collected together for reference or analysis. And so our hope is that the data, whether it be height or coat color or whatever it is, reflects reality in nature. It reflects the true, or as close as we can get to the true underlying sort of phenomena. In this case, in order to collect data, or we can refer in some cases in this lab to variables. A variable is any characteristic, number, or quantity that can be measured or counted. And variables can come in different forms. I will explain in more detail what a variable is in the next lab, because in that lab we'll talk about the analysis of data and the use of some like really simple statistics that are implemented in Microsoft Excel. Okay, so in order to acquire this data that is the central tenet of a scientific investigation, we need to use measurement. Measurement being the process of associating a number or a set of numbers with physical quantities and phenomena. Now, we sort of take this for granted, I think, both in society and in some scientific disciplines, actually. But measurement is really at the core of all science. If we can't measure something and determine accuracy and precision, we're not really doing anything. And so, I mean, really think about this. When we refer to parent height or offspring height, we're kind of referring to the measurement, right? We kind of have an understanding that usually we measure height as feet and inches. We just saw the example of the distribution of heights in men and women as feet and inches. So that number, 5 feet 11 inches or 5 feet 6 inches, is a number. It's some quantity. It's a distance that is associated with the amount of material that makes up your body from your head to your toes. Okay. We can get into the philosophy of measurement and measurement theory and that kind of thing, but that's a little bit outside of the scope of this. But this is the kind of like the core of what we're doing. Data is power. You have to understand that like data is incredibly valuable for understanding the world that you live in, right? Your smartphone, the GPS, the internet, anything that we value, many of the things that we value ultimately start here, right? When you're using Google Maps, it's measuring your physical location, right? Those are data. And it turns out that companies like Google are actually selling the data. But anyway, that's a different discussion. All right. So let's see. Okay. Let's come back to this. For some reason, it's not letting me go. All right. So for each of these questions, we have different variables of interest. So again, how does parent height influence offspring height, etc.? Well, we need the measured height of offspring. That'll be like student height, measured in centimeters, cm, centimeters. Units are very important here because you can measure height in feet and inches. You can measure it in millimeters. You can measure it in whatever. We're going to use centimeters. And then the reported height of the biological mother and father of the offspring. I listed this as reported height because we're not actually measuring, and the people who have measured this previously for this course over the last few years haven't actually measured the height of their mother and father in centimeters. They just said, how tall are you? I'm 5'4", I'm 5'11", I'm whatever. We then have a calculated variable, which is not something that we actually measured. It's kind of like a mathematical hypothetical thing, which is the average parental height. So the average parental height is calculated from this reported measurement. That is the height of the mother plus the height of the father divided by 2, because there's 2 individuals. For the second question, what is the effect of temperature on limb proportions? We have a couple variables. We have the latitude of ancestral populations as a kind of rough proxy for temperature in degrees. And then seated height is another measurement or variable that's a proxy for torso length, right? So sitting in a chair and measuring the height of basically your height while you're seated. We can then calculate relative leg length as the overall student height divided by seated height. So it's a proxy for how long the legs are relative to total height. And so it's a ratio. This number divided by this number is a ratio that can range from 0, hypothetically, to infinity. In humans, it's not going to range from 0 to infinity, but rather from about 0.43 to 0.54. And so in one sample, that was the range, the minimum and maximum values for that ratio. The mean is 0.5, which means that the legs are about half of height, 50% of height, with a standard deviation of 0.02. And I'll talk about what standard deviation is next time, but standard deviation is basically the average difference between each individual's real relative leg length and the sample average. How much each individual differs from this average. It's kind of like a measure of, you can almost think of it as like a measure of variation in the sample. Okay, number 3, how do body size and proportions influence grip strength? And some of the variables that have been collected previously are forearm length in centimeters, maximum hand length, maximum palm length, and grip strength in pounds, using like a portable kind of like spring-loaded grip strength where you pull as hard as you can and you measure how far the needle goes, which represents pounds of pressure. These were included more recently, just to see, out of curiosity, if like an individual has relatively longer fingers, does it influence how strong they are? And so from this we can calculate relative forearm length, which is the length of the forearm divided by height. And we do that because taller individuals will have longer forearms, because there's a positive relationship or correlation between arm length and height. Now, individuals of the same height may differ in forearm length naturally. Some individuals have arms that are a little bit longer. Some individuals have arms that are a little bit shorter. So we can standardize forearm length by individual height, so that the thing we're looking at is not the effect of how tall they are necessarily, but the length of their forearm. We can then look at relative finger length as your overall maximum hand length minus palm length, standardized by height, divided by height. And so in case you missed it, or if you're still confused about this, we're not going to be measuring this on ourselves. I have a data set that I'll be sharing with everybody that includes data that were collected previously over multiple different classes at Hunter. So that's really cool, because we have a data set of like 200 individuals or something that have taken this course over many different semesters. And so we'll use that. Okay, so for latitude, this is what we're referring to. We're referring to latitude in degrees, basically from north to south. And so high latitude places are places that are really cold usually. The lower the latitude gets to 0, gets closer to the equator. Anything past that in the negative direction is warm until you get to, hey, guess what? The cold place again. So that's latitude. And we're thinking of latitude of like ancestral, like what one might think of as an ancestral population. So for me, I'm like a generic European person. That's really not a surprise. But so like maybe there's some German in there, there's some Polish in there, things of that nature. And so there are websites, and this is one in particular called latlong.net, where you can type a place in. We'll say Germany. You can see I've already done it. And find that apparently I'm a bot. So let's go back to this. So the latitude here for Germany would be 51.16, So we could just call that 51.17 or 51.2 for our purposes. So that's what that looks like. And so here's the data set that I'll upload to Blackboard. And so today, like, you'll be able to look at this. You don't have to do anything with this right now. I will give you more information next lecture, next lab, for how to like start processing this and analyzing it for the lab report. And I'll explain what the lab report is. I'll give you a template where you fill in the information. It's a 2-page thing. It's really not so bad actually. All right, so this is Microsoft Excel. Excel is a commonly used spreadsheet for collecting data and analyzing data as a spreadsheet software. You can also use Google. Google has something, I'm forgetting what it's called, but it's like their equivalent to Microsoft Excel. And so usually each column, right? So you have columns A, B, C, D, E, right? And so on and so forth. Each column is usually the variable of interest. So we have a height column in centimeters. We have a mother's height column. We have a father's height column. We have these other calculated columns that I'll talk about in just a second. And so we have everything here, right? So latitude, seated height, forearm length, hand length, palm length, and grip strength. Now you'll see that many of these are missing values. That's because these classes didn't collect those data. This is something that I started doing last year just out of my own curiosity. All right, so in a spreadsheet like this, when you're collecting data, each column is a variable. It's a thing that you're interested in. Each row is an individual. So this is individual 1, individual 2, and so on. And this is individual 1 is 165 centimeters tall, individual 2 is 173 centimeters tall, and so on. Of course, this doesn't have individual identifying information in it because that would not be cool, right? We don't want to share information about somebody's height, even though it's a fairly minor kind of thing. And so we'll obviously use anonymous data. So we can just scroll through and see, let's see, how many individuals? 170 individuals, because the first row is the labels. 170 individuals. That's a pretty decent sample. This is not a random sample because it's college students from Hunter, but it's still kind of cool nonetheless. Okay, so if I scroll over to this cell, each of these things is referred to as a cell. If I refer to this cell, D2, at the top of the screen, you'll see this window here. So this is the average parent height, which is a calculated variable, as I mentioned before. It's calculated because there's nowhere where you can measure the average height of your biological parents. So to do this in Excel, and this is already done for you, but I just want to show you, you type equal, and Excel has built-in functions. So if you type in average, right, you can see that there's a function called average, and it does nothing because we have to put numbers in parentheses after that. And so we could do the average of, let's say, 5 and 5. It's going to be 5 because 5 plus 5 is 10. Divided by 2 is 5, right? So we can type equal average and scroll over to include different cells. So there's the average height for these two individuals. 163 plus 163 divided by 2 is 163.5. And we can take that column and just drag that equation across the rest of the data set. And so as you go through, you can see that it automatically moves these cells down with the target cell. So we've calculated average parent height. We've calculated seated height divided by standing height. So seated height divided by the student's height. And we've calculated forearm length divided by height, to standardize by height. What else? Let's see. We only have, from my last summer class, 15 individuals. But this measurement is palm length divided by hand length. We could, I think I mentioned it as palm length, let's see, hand length minus palm length. That will give you the length of the fingers in centimeters. And then we can standardize this by the height of the individual. So basically what this is saying is that this individual has fingers, has like the longest digit that is about 5% of their height in centimeters. So this individual has fingers that are like 2% of their height, right? So if you're just looking at the numbers here, it ranges from 0.02 to about 0.05 if we round up. In order to make this equivalent to like a percent, you would just multiply it by 100. So this is 4.59%. We can just call it 5%. We're not going to multiply it by 100. We're just going to keep it in the original units. And that's it. I'm going to post this on Blackboard so you can see everything. So you have it there. I'll post the lab template as well. You don't have to do anything. There's no like lab worksheet 1.2. But I'll let you know if we'll do anything prior to the next lab report. So that's it. I hope everybody's doing well, and I'll talk to you next time.